We are going to briefly discuss the 3 stages of embryonic development. Embryonic development begins as soon as fertilization occurs. In animals, fertilization refers to the internal reproduction using eggs and sperm, both being haploid or 1N to form a diploid or 2N organism. 2N refers to chromosome copy number that we've discussed before. In embryonic development of any animal, we look at 3 major stages: cleavage, gastrulation, and organogenesis. We'll look at each individually and their main products of each phase. Cleavage is simply division. In this case of embryonic development, we look at very rapid cell division. So we start with a single cell, a fertilized egg, and we're going to cut it in half, and cut those in half, and cut those in half, and so on. Now, under normal cell division, you might be aware that there is a significant period of time associated with growth of the cell. We don't see that in embryonic development. This division happens so quickly that the cells don't have time to grow properly in between. So with every division, the cells get smaller and smaller. This is okay. We'll give the cells time to grow. Obviously, you're not still the same size as you were as a fertilized egg. Remember, fertilized egg is also known as a zygote, right? So we want to take that zygote, that fertilized egg, and produce something known as a morula. This is just a solid ball of cells. Eventually, we're going to divide that cell enough. We're going to have so many cells that that property that we know as cell adhesion will no longer come into play. Cell adhesion means cells want to stick together. They want to be close to one another. But when you divide these cells so rapidly, they eventually are going to pull apart and form what we call a blastula. This blastula you can think about like a soccer ball. It's going to have cells around the outside, but it's going to be hollow in the middle and eventually fill with fluid. This fluid is cytoplasm, or the basic equivalent of cytoplasm. Cytoplasm, remember, is the filling in all of our cells. So as these cells divide, they actually are forcing out some of the cytoplasm because they get smaller and smaller and smaller. In my diagram here, we start with that fertilized zygote, 2N, that was produced as the union of sperm and egg, and we divide and divide and divide until we get so many in this solid ball of cells that we called the morula. We then produce this blastula, or a hollow ball of cells. Notice this word here, blastocele. C-O-E-L is cavity. So essentially what we've produced is a ball of cells with a cavity in the middle. And we said this cavity is actually going to be fluid filled. This becomes important for producing and providing nutrients for our cells as they grow so fast. This is the embryo early stages of a frog, and you can see in the cleavage stages we really are just dividing cells. You notice they're not all the same size, they're certainly not all the same shape. All we're looking to do is to very rapidly produce a very large number of cells.